Here we are starting the section 5.8, prokaryotic reproduction and survival. At this point, we've learned quite a bit about prokaryotes and their structures. Now we're going to figure out, how they make copies of those, pass them on to their offspring, and then how, and that's pretty easy, honestly, with prokaryotes. And then we'll also talk about ways they can survive very harsh conditions. As far as reproduction goes, most prokaryotes are going to undergo binary fission. In times where they can't undergo binary fission, when times are harsh, they might produce endospores. When times are plentiful, they might produce a kinetase. Binary fission is very similar to mitosis that you learned about last semester. Now, it's not mitosis because mitosis is the division of the nucleus, the nuclear material in the nucleus. We're looking at binary fission, which is a lot more like cytokinesis, where you're splitting the cellular contents, but you're also making a direct copy of the nucleic acids as you're doing it. So you're splitting into two distinct cells after having made a copy of the circular DNA of the bacteria. When times are plentiful, cells can produce what are called a kinetase. A kinetase are large food-filled cells. Basically, the bacteria are creating their very own refrigerator, except it's not actually keeping something cold. It's like a pantry where if you have extra food, you can store it for later. I don't know, think like 1960s bomb shelters. You got lots of stuff right now, put it into a shelter so that you could have it when times are difficult during, I don't know, post-apocalyptic scenario. One cell in a group of cells will become enlarged and filled with vacuoles that hold extra food. And then as times get worse and there's not food around, it begins to pump that food out to the other cells in the chain. When times get really bad and you've exhausted, or the bacteria, the prokaryotes exhausted all of its other venues, all of its other food, it might produce an endospore. Endospores are these sort of large, very hard structures that form inside the bacterial membrane. They surround the nucleic acids, the nuclear material, and the bare minimum needs for keeping the bacteria alive. And that way, when the rest of the cell dies, that spore, that hardened structure, goes into a state of stasis, and it just sort of stays dormant until conditions become good again. This is why, I don't know, if you're sick with a bacterial infection and you cough all over your pillow, that bacteria does not survive long on your pillow, but it might go into this endospore structure where it will stay dormant until it's reintroduced into a nice, happy environment. Maybe, I don't know, your partner's lungs if they lay on the same pillow after you had laid on it. And that will open up once it gets into their lungs, and a bacterial infection can restart. Bacteria need different energy requirements. They've had so long to evolve that basically, if there's something out there, a bacteria can digest it. Now, bacteria can be autotrophic or heterotrophic. Further, that can be broken down into chemoautotrophic and photoautotrophic, or chemoheterotrophic and chemoautotrophic. So basically, think of absolutely anything you can, and bacteria can get food from it. Chemoautotrophs are going to take inorganic materials from their environment, reduce them. So basically, you're going to use the energy from the electrons in them in order to convert those inorganic materials in the environment to organic materials. And the other side of that coin is photoautotrophs. Photoautotrophs are going to use the energy from light to produce organic materials from inorganic materials. So your famous photoautotrophs are plants. Plants take light energy to take inorganic carbon dioxide and convert that into an organic form, bound in sugar. So here we see an example of a chemoautotroph. This is at a black smoker down at the very bottom of the sea. They're using the energy from inorganic molecular bonds to form organic molecules. So you get a lot of inorganic materials spewing out of that vent. The bacteria that live around there are using those materials that are spewing out of the vent to create their own organic molecules. And here's an example of a photoautotroph using the energy from light in order to form chemical bonds of sugar. So these are some algae that are working very hard to do that. As far as heterotrophs go, you got photoheterotrophs and chemoheterotrophs. Just like we had two sides of the autotrophs, we got two sides of the heterotrophs. For photoautotrophs, there we have plants. I'm sorry, we don't have plants. Plants are photoautotrophs. There we have organisms that are going to use light to generate ATP, but they still need to take in organic molecules from the environment. So they cannot fix their own inorganic molecules into organic molecules. They need to take in organic molecules from the environment, and they can get that oftentimes from the photoautotrophs or from the chemoautotrophs. So photoheterotrophs, they get energy from light, but they can't necessarily create their own organic molecules. And then chemoheterotrophs, I've got a bacteria on here, but they're just like you. They're going to use energy from organic bonds in order to form new organic bonds. So they're not building anything at all. They're just using the material in their environment. They're getting energy from it and building their own molecular organic molecules from that. Bacteria and prokaryotes have different types of oxygen requirements. They can be described in one of four ways. They could be obligate aerobes, aerotolerant anaerobes, facultative anaerobes, or obligate anaerobes. Obligate aerobes absolutely require oxygen to survive. Without oxygen, the obligate aerobes will die. So obligate means they're obligated to have oxygen. Aerotolerant aerobes don't necessarily use oxygen, but they're not poisoned by it. And you can imagine why an organism would be poisoned by oxygen. It's extremely electronegative. It steals electrons. So aerotolerant aerobes don't use oxygen, but they are not poisoned by it. They can grow just fine. An example of this would be, say, probably tube 4-ish, where they're growing at the top and in the middle, or even tube 5, they're growing throughout. Facultative anaerobes are amazing because they can undergo fermentation when oxygen is not present, but when oxygen is present, they can undergo aerobic cellular respiration and produce a lot of ATP. They can grow everywhere. These guys are very, very adaptable to situations. Your muscle cells are facultative no, they're not facultative anaerobes. They are facultative cells that can switch on and off. Facultative anaerobes can grow in both conditions and do an amazing job. And then obligate anaerobes, the obligate, like we said before, these guys require anaerobic, no oxygen. So if oxygen is present, they die. They are highly reactive to oxygen that steals electrons. Organisms don't just have oxygen requirements, though. They have nitrogen requirements. They have carbon requirements. Nitrogen is very important because nitrogen is used to build amino acids. Nitrogen is an important part of ammonia, which is in turn important for amino acid building, which makes proteins. Plants and animals cannot take nitrogen out of the environment and fix it and put it into into an organic form. But prokaryotes can, and that's kind of a big difference. That's a big distinction, is prokaryotes can fix nitrogen. They remove nitrogen from its gaseous state and put it into an organic molecule. Nitrogen fixers are bacteria that oftentimes live in conjunction with or in symbiosis with plants. They live near the plant's roots. They will take atmospheric nitrogen and turn it into ammonia. And then in turn, that ammonia can be turned into amino acids. And then those amino acids that the bacteria produce are used by plants to produce proteins. That's how we would get our nitrogen, is eating plants, not by breathing it in. We don't get anything from breathing nitrogen in. We have to take nitrogen in with our diet in order to process it to build amino acids and proteins. So plants and bacteria tend to have symbiotic relationships. Plants need ammonia to produce proteins, and bacteria can produce that ammonia that are then broken down by proteins that are produced by the plants. And then the plant takes it up. If the bacteria gets sort of a nice place to live in the roots of the plant, sort of a safe environment, and the plant might even donate some nutrients to the bacteria to keep them healthy, and then the bacteria will give the plant the ammonia it needs in order to produce amino acids. Some bacteria who are these nitrogen fixing bacteria might have heterocysts. Heterocysts are cells sort of like they have the same structure that looks like in a kinete, where they're just sort of enlarged cells that create low oxygen conditions within them. They literally pump oxygen out in order to encourage more nitrogen fixation. So they have very, very thick cell walls, very slow oxygen diffusion, and that allows for more efficient nitrogen fixation to occur.